Assalamualaikum and hi. We are from Group 6. We present to you about our project HIV Vaccines. I am Nur Atira Muhammad Zaki and my team member will explain to you about the HIV vaccine development. HIV vaccine. Introduction to HIV. HIV is a virus that causes the weakening of the immune system leading to a fatal disease. AIDS that stands for Acute Immunodeficiency Syndrome. HIV stands for immunodeficiency virus that attack the white blood cell of the body. HIV is transmitted through the blood and urogenital fluids. HIV is the cause of the AIDS. AIDS is left untreated. A group of symptoms of appear in the effect person who is no longer able to combat the infection. The virus at first disrupts the macrophage and T helper cells and then other white blood cells. Genetic material in HIV is double-stranded, which is RNA. This RNA is converted to DNA by the reverse transcription enzyme found in HIV. The virus is roughly spherical and is about 100 nanometer in diameter. It has an outermost layer called envelope, which is made up of lipoprotein. These are spike-like structure. Its surface made up of glycoprotein. The genetic material is enclosed in the capsid. There are 9 genes encoded by the genome of HIV. This is a structure of HIV, which the lipoprotein outermost layer is made out of the host cell membrane, which protect the virus from the attack of the host immune system. The host immune system cannot distinguish between the virus and its own cell at this stage. The function of reverse transcriptase is to form the DNA to mRNA. This DNA form is integrated into the host genome by enzyme integrase. That protease enzyme make the virus function. HIV cycle. HIV is a copy or replicated in the human genomic DNA by integrating its genetic material into DNA of the host. Only the genetic material RNA in HIV enter the host cell and not the protein code. Step in HIV replication is the fusion of viral envelope with the plasma membrane. That the DP102O of the envelope is complementary to the CD4 receptor of the macrophage. The capsid enters the human cells. Protease break the capsid so that the RNA gets released into the cytoplasm of the host cell. The RNA is converted to complementary DNA by the action of reverse transcription enzyme. This cDNA is converted to double-stranded DNA. This double-stranded DNA is integrated into the genome of the host cell by the action of the enzyme integrase. Then the DNA will undergo the transcription to form viral mRNA. The protein will perform infect T helper cells. The aim of the HIV vaccine is to reduce viral load of prevent infection through the induction of humoral and adaptive immune responses. It is still an enigma that the vaccine are not able to induce HIV envelope of the evade the adaptive immune response. The vaccine is not able to elicit the humoral response that is broadly reactive, neutralizing the antibodies. In the envelope, protein of HIV leads to evading the immune responses. Hence, the vaccine need to combat high degree and diversity of antibodies against the HIV.
The first step to develop a vaccine is to extract the DNA. But you might ask yourself, what is extract DNA? DNA extraction is a method to purify DNA by using physical and or chemical methods from a sample separation DNA from cell membranes, proteins, and other cellular components. In this project, we extract DNA from the blood using magnetic beads, which is the newest method of extracting DNA. Whole blood DNA isolation using magnetic beads works by capturing DNA on magnetic beads coated with the matrix of silica for binding nucleic acid. The whole blood cell first must be lysed using SDS or similar detergents. The next step involves a mixture of the lysed cells and magnetic beads, allowing DNA to bind the beads. Several rounds of washing in the presence of a magnetic field then separates the DNA captured on magnetic beads from other unbound cellular contaminants. Then, a low salt buffer elutes the DNA from the beads.
Next, I will explain about PCR amplification of the gene and plasmid vector. So, PCR amplification is a technique used to amplify a segment of target gene and produce a large number of copies by using polymerase chain reaction. The materials that are used in PCR amplification of genes are nucleotide upstream primer with 3' GGAT, CCGA, TTG, TCA 5', downstream primer 5' CGTAA, CCA, TCG, TTAAG 3', and the cofactor magnesium chloride, 1 unit of tag polymerase, 10 times VR buffer, sterile water, and 10 mn DNTP. Next is the steps in PCR amplification. The first step, which is step 1, is denaturation. In this step, we heat the reaction strongly at 96 degrees to break the hydrogen bonds holding between complementary base pair of the DNA strand. This provides a single-stranded template for the next step. Step 2 is annealing. So, cool the reaction to the temperature about 55 to 65 degrees Celsius in order to let the primer bind to their complementary sequence to the single-stranded template DNA. And lastly, step 3, which is extension. We raise the reaction temperature to 72 degrees Celsius so the tag polymerase can extend the primers and synthesize new strands of DNA. So, to make it more detailed, the PCR employ the enzyme DNA polymerase, which guides the synthesis of DNA on a single-stranded DNA template from deoxynucleotide substrates. So when annealed to a longer template DNA polymerase adds nucleotide to the 3' end of a custom-designed oligonucleotide. Thus, if a synthetic oligonucleotide is annealed to a single-stranded template containing a complementary area to the oligonucleotide, DNA polymerase may utilize the oligonucleotide as a primer and elongate its 3' end to form an extended length of a double-stranded DNA. So you can see in the diagram for the polymerization reaction, which is we have the original DNA to be replicated and are expand according to the steps. So these cycles is repeated 25 to 35 times, which take 2 to 4 hours, depending on the length of the DNA area to be copied. Next, is the PCR amplification of plasmid. So the material that are used, which is all the same as the material as amplification of the gene, but with the added of restriction enzyme. Before the PCR amplification, the plasmid vector has been cut with the restriction enzyme. So in this case, the plasmid vector used is PUC90. This process of PCR amplification of plasmid vector is the same as PCR amplification of gene, which are denaturing, annealing, and extension. That's all for PCR amplification.
Purification of DNA. Two general procedures that can be used in purification are centrifugation and chemical extraction. By centrifugation, the sample is spun at high speed and larger components will be precipitated to the bottom of the tube. The unwanted particles that remain in solution are removed. The precipitate in the tube is redissolved by adding water. Phenol is added to the tube to dissolve and remove most of the protein. When phenol is added to water, the two layers of solution are formed. Upper layer is water and the lower layer is phenol. When the mixture is shaken, two layers mix temporarily, and when they stop shaking, the layers separate again. The protein will be dissolved in phenol, but the DNA and RNA stay in the water layer. The water which contains DNA and RNA is sucked off and stored into another test tube. To get rid of the RNA, ribonuclease is added to the test tube, and it will convert RNA into tiny fragments but leave the DNA unchanged. Then, alcohol will be added to the test tube. It will occupy all of the water and excrete the larger and less soluble DNA out of the solution, while small RNA fragments remain dissolved in the solution. The centrifugation is repeated and DNA is sedimented to the bottom of the test tube. The solution that contains RNA fragments is poured off.
The next step to develop vaccine is separation of DNA. In this case, to separate DNA fragments, the gel electrophoresis technique is used. So, what is gel electrophoresis? Gel electrophoresis is a method used in laboratories to separate charged molecules such as DNA, RNA, and proteins based on their size. An electric current is passed through the gel, causing one end to be positively charged and the other to be negatively charged. How is gel electrophoresis carried out? It is accomplished through the use of a few procedures. First, we must prepare the gel. Agarose gels are commonly used to visualize the DNA fragments. Following that, we prepare the DNA for electrophoresis. Prior to electrophoresis, a dye is added to the DNA sample to make it more viscous, preventing it from floating out of the wells and allowing the migration of the sample through the gel to be observed. After that, we separate the fragments. The electrical current is then activated, causing the negatively charged DNA to travel through the gel and toward the gel's positive side. The final step is to visualize the results. The electrical current is turned off and the gel is withdrawn from the electrophoresis tank once the DNA has gone sufficiently far across the gel.
Okay, next I will present about choosing a vector. First, we need to know what is clone vector. During cloning, a cloning vector is a small piece of DNA that can be inserted into an organism for cloning purpose but remain stable in the organism. An organism cell, a virus DNA, or a bacteria plasmid can serve as the cloning vector. By incorporating insertion sites into the vector enables the convenient insertion and removal of DNA fragments using a restriction enzyme to cut the DNA. Vector DNA and foreign DNA with compatible ends can be joined together by molecular ligation. The DNA fragment resulting from the restriction enzyme treatment contain either blunt ends or overhangs called sticky ends, making molecular ligation the only method of joining DNA fragments. Subcloning of DNA fragments into a vector designed for more specific use can be done after it has been cloned into a cloning vector. Next, the criteria for vector is its reasonably small and manageable DNA molecule. Second, moving the vector from cell to cell should be easy, and the last is growing and purifying large amounts of vector DNA should be straightforward. Why the vector is important? Because the importance of choosing a vector is vector can detect the presence of the vector. We also can directly select for cells that contain the vector and insert genes into the vector, then detect the presence of inserted DNA in the vector. Next, I will explain about the step in clone vector. We have 5 steps, which is isolation, selection, incorporation, transformation, and recombine. First, first step is isolation of donor DNA fragment or gene. The first thing to do is to isolate a DNA fragment from a donor. It is possible to isolate desired genes or DNA fragment using 2 different methods. Gene cloning is facilitated by restriction of the endonuclease enzyme, which is a key enzyme in the process. The action of this compound is treated to a specific site. By using restriction enzyme, we have 2, which is BAM, HI, and ECORI. You can generate either blunt-ended or sticky-ended DNA fragments. Synthesis of complementary DNA strand using reverse transcriptase enzyme. reverse transcription enzymes, which is synthesis complementary DNA strands for mRNA of the desired gene. Next is a selection of suitable cloning vector. An isolated gene cannot replicate itself when incorporated into a host cell, so a donor DNA fragment will not replicate in host cell. The vector should be selected before the donor fragments are introduced into the host cell. Inside the host cell, cloning vectors can self-replicate their DNA. Cloning DNA fragment replicates inside the host cell with the help of the cloning vector. In here, we use the plasmid as a cloning vector. Cloning vector must be self-replicating inside the host cell in order to function as cloning vectors. Also, donor DNA must not interfere with the vector's replication property when introduced at the restriction site for restriction enzyme. Furthermore, the gene must be encoding a marker that can be used to identify recombinant cell letters on. Next is about incorporation of donor DNA fragment with plasmid vector. Using the same restriction enzyme that is used to isolate the donor DNA fragments, the plasmid vector is cut open. We mix fragments of donor DNA with plasmid vectors, and recombinant vectors are formed by base pairing donor DNA fragments and plasmid vector in the presence of DNA ligase. And here you can see the structure, how we cut the enzyme using the restriction enzyme, and then fragment, it have a single stranded overhangs, and fragment with matching overhang base pair and stick together, and DNA ligase will seal the gaps. Okay, next step is transformation of recombinant into vector suitable host. An appropriate host cell is transformed with the recombinant vector, in other words, bacteria cell. Recombinant vectors are automatically taken up by some bacteria since they are naturally transformable. But bacteria that are not naturally competent recombinant vectors are incorporated artificially through methods like calcium++ ion treatment and electroporation. Okay, the last step is isolation of recombinant cell. In culture media, recombinant host cells are grown alongside non-recombinant host cells, with the latter multiplying as the recombinant cell is grown. A marker gene on a plasmid vector is used to distinguish recombinant cells from non-recombinant cells. It provides a platform to make recombinant cells that are sensitive to ampicillin. For instance, we use PUC19 vector contain both ampicillin and tetracycline resistant genes. It used to draw out the ampicillin resistant gene from the plasmid by using our restriction enzyme. Yeah, you can see the process how we do the clone vector.
Effective HIV treatment and prevention options are increasingly available due to the limit, accessibility and other treatment challenges. The urgent need for an HIV vaccine remains. Competing strategy emphasis immunologic. HIV will likely necessitate a application of reinforcing immunologic mechanism for success. As vaccine technology and clinical trial network develop to combat HIV were leveraged in the other vaccine response. Important to efficacy trials are underway, and innovative HIV vaccine design approach have yielded a robust and diverse pipeline of early stage with the potential to incorporate next generation adjuvants. The stage is set for HIV vaccine developers to move this concept forward together to achieve the unlimited public health tool and effective HIV vaccine.